Thank you so much, ma'am. And continuing, our first speaker is Dr. Raghavan Varadarajan from Indian Institute of Science, Bangalore. And I request our moderator to introduce him.
Thank you. I've checked that Raghavan doesn't mind in the first place saying anything or nothing about him, so I have freedom to do that. But still, he's a professor at Indian Institute of Science in the molecular biophysics unit, and he's been working at the intersection of biology and physics for a long, long time. He's one of the very young, not very young any longer, but he became full professor at a very young age. And his work on, even on SARS-CoV-2, which hasn't been mentioned here, but in the context of vaccine manufacturing, vaccine design and development has been very, very remarkable. He has also worked on flu. Raghavan.
Great. Thank you, Vineeta. And a pleasure to be here. That was me a long time ago, as you can make out. Now, I'm not an immunologist, so I apologize in advance for any mistakes and omissions. One of the reasons is there's so much jargon and terminology in immunology that I just forget. There are more cytokines and T cells than there are institutes in Delhi, which is saying something. Anyway, so let me start. Next slide, please. Okay. So just a few definitions. I'll talk about pandemics and vaccines to combat pandemic threats. So, all of this you probably know, but good to just get started. So if something is endemic, it's found within a specific geographic area, and it remains confined largely to that region. An epidemic is an outbreak, which is again localized to particular regions of the world. And a pandemic is one which occurs over a much wider geographical area, and it can affect a significant proportion of the world's population. And then, like what happened recently with COVID-19, you have a public health emergency of international concern, which triggers a bunch of restrictions to be put in place. And then there's certain diseases which, even though they may or may not have caused a pandemic yet, are of great concern. Next slide, please. Okay. So pandemics are not new. There have been pandemics throughout history. So there have been pandemics throughout history, and not all pandemics are viral pandemics. You can see the Black Death or the bubonic plague, which occurred over, a very long period of time, has killed almost 200 million people. Okay, and down to SARS-CoV-2, which has probably killed a few million, 7 or 8 million people till date. And we've had other pandemics, within the past 100 years, HIV, AIDS, COVID. There have been multiple influenza pandemics. So these are things which happen with some frequency. Okay. So what might cause the next pandemic? Again, people don't really have an answer, but more likely than not, it's going to come from some kind of zoonotic event, which results in transfer of a pathogen from outside humans into humans. And likely, if it's a pandemic virus, it's going to spread through the air, because as we have seen in the case of COVID-19, once you have human-to-human air transmission, it's impossible to stop by all the restrictions that were put in place. Now, while it's conceivable that you could have pandemics caused by viruses which don't have respiratory transmission, it's probably not so likely. So now this is the extent of my knowledge of a lot of immunology, and you've heard a lot about this. So you have innate immunity, and then if that doesn't if the pathogen escapes, then you have adaptive immunity in the form of B lymphocytes and T lymphocytes. Now, in terms of vaccines, right? So you have nucleic acid vaccines, and these can be DNA or RNA vaccines. And these are relatively safe and easy to develop. In the case of RNA vaccines, temperature storage remains a consideration. So the early RNA vaccines that we had during the pandemic required ultra low temperature storage, and even the ones that are administered now still require refrigeration at frozen temperatures prior to being deployed. And this can be a constraint in many parts of the world, which is why one of the reasons why mRNA vaccines, despite being so efficacious, were not so widely deployed in terms of geographic areas. DNA vaccines are much easier to manufacture and to store, but their immunogenicity is low, and they really have not been particularly successful. Then you have viral vector vaccines, which is what most of us received. In our case, an adenovirus encoding the spike. And these can either be replicating viral vectors or non-replicating viral vectors. And so the non-replicating viral vectors go inside cells, and they contain a gene which is the important antigen on the infectious organism. And so then those cells produce that protein, which in turn generates an immune response. Okay. And then you have the virus itself, which can act as a vaccine. And that's been, for a large number of viral diseases, having an inactivated virus has proved a successful modality to combat the disease. One of the problems with having inactivated virus is if you have a new pathogen which requires high containment levels, then manufacture at scale of such kinds of modalities becomes very difficult. Another probably less appreciated issue with this kind of modality is that the way many viruses infect cells, the SARS-CoV-2 as an example, is that they have on their surface a protein or proteins which are responsible for binding to a cellular receptor and for, at some point, either during entry or after entry, in causing fusion of the viral and some cellular membrane, which eventually results in the release of the viral contents into the cell. Now, one of the features of such viral surface proteins, of many of these viral surface proteins, is that they tend to be what are called metastable. So that is the form that is located on the surface of the virus, the so-called native form, is not the most thermodynamically stable state of the protein, because the energy that it uses to get to the most stable state is used to overcome the barrier which is present when two membranes have to fuse. In this case, the viral membrane and some part of the cell, either the plasma membrane or the endosomal membrane or whatever. Now, because the protein is metastable, very often during conditions of production, if one is not careful, the protein can transition to a state which happens after fusion, and that state is called the post-fusion state. And that's useless from a vaccine point of view, because the antibodies that are elicited against the surface protein have to be able to bind to the native form of the surface protein and prevent infection into cells. Okay. And then, of course, one can have live attenuated virus. This, again, for a new pathogen is time consuming. You have to attenuate it to exactly the right degree so that it gives a protective immune response, but does not cause severe infection, including in people whose immunity may not be very strong. So, which is why in many cases, although live attenuated viral vaccines have been around, they are not always, I mean, there are, of course, exceptions, like in the case of polio, but they are. And then there are protein subunit vaccines, in which you take you have to choose the right protein from the virus, typically a surface protein, express in recombinant form, make sure it's in the right conformation, and purify it. And then typically these need to be combined with immune enhancing molecules called adjuvants. And then these are injected into people. You could also have virus-like particles which contain some viral proteins, including the surface proteins, but lack others, and they're non-infectious. So these are both very safe modalities. Now, how long does it take to develop a vaccine, and what are the different kinds of steps? The first step, and the most difficult often, is the exploratory research, which can take years, or in the case of difficult viruses like HIV, for which we don't have a vaccine, it can take many, many decades before you are able to find a solution, if one exists. And then one needs to carry out preclinical safety and efficacy studies of a vaccine formulation typically in small animals, and to check to see that they elicit the desired immune responses. And then one carries out clinical trials. The initial phase 1 trial is used essentially to look at the safety of the vaccine, whether it's producing adverse immune reactions or unpleasant side effects, and so on and so forth. And then one looks at both the safety as well as the immunogenicity in a phase 2 trial, and then you actually look at the efficacy in a phase 3 trial. So earlier this would take of the order of a decade, and there were various reasons for that. Some of them were difficulties in knowing what antigen to use or what immunogen to use, and others were just in the regulatory procedures. Now, during the pandemic, all these timelines have been greatly compressed, and within a 1-year period, one could have an effective vaccine. Of course, this will not be true in all cases, but this is what happened. But even 1 year, as it turns out, was too long to prevent, I would say, the majority of the world's population from being infected. So we need to be able to make vaccines much faster than that. So if one looks, in the case of COVID, if you look at the map of vaccinations all over the world, so with darker colors showing, more vaccination being done as a percentage of the population, you can see that vaccination coverage was highly non-uniform. And even though with all the advancements that had been made and all the accelerated timelines, it failed to reach many of those who were in need before they were infected. And we have seen that in this country also. Okay. Now, what are some of the features that are important in understanding in the process of vaccine design? So again, all of you are familiar with all of this stuff. So now just here, something that you should be but may not be familiar with is how do you quantify the stability of a protein? So the stability of the protein is quantitatively described in terms of the free energy difference between the unfolded and the folded states of the protein. Okay? And why is this relevant for vaccine design? You need a protein to be sufficiently stable such that when it's injected into the body, what the immune system sees is the folded state and not a misfolded or the unfolded state of the protein. And of course, it has to be stable to conditions of storage and transport and so on and so forth. Okay. And for monomeric proteins, this is a function of temperature. And in fact, interestingly, it shows this so-called bell-shaped dependence on temperature, where stability is reduced at both low and high temperatures for reasons I don't have time to go into. Okay. So an immunogen is a component of the vaccine that stimulates the immune system to produce, well, antibodies as well as cellular responses against it. And neutralizing antibodies are ones that bind to the virus. I'll be talking largely about viruses here. And they prevent infection. And the ability of a vaccine to elicit neutralizing antibodies is critical to its protective efficacy. And very often, the neutralizing antibody titers are a correlate of protection. So folded proteins, although you might see all these beautiful pictures, they are not static structures. They undergo all kinds of conformational fluctuations. And when they do, this can lead to the exposure of irrelevant conformations. And often these can act as decoys to the immune system. And this happens in the case of HIV. So where you get lots of antibodies to the surface protein, but they are not neutralizing. And a failure to elicit neutralizing or poorly neutralizing antibodies can be very detrimental, because in some cases, non-neutralizing antibodies can actually enhance infection, as has been seen for dengue. So protein stabilization through, among other methods, genetic engineering can enhance the elicitation of neutralizing antibodies and thus vaccine efficacy. And this is true of all kinds of vaccine formulations, including, as I will briefly mention, the mRNA vaccine formulations that were so critical in combating COVID-19 in many parts of the world. Okay. So very often, designing the right immunogen can be the rate-limiting step in vaccine development. If you don't have, that's, it's one of the early steps, but if you don't have that right, you can't move forward. And this is true pretty much in virtually any case. So protein stabilization can enhance the production of neutralizing antibodies and thus vaccine efficacy. So this is just a diagrammatic example. Supposing we have a protein which is largely has a significant population of misfolded or unfolded protein along with it. So when that is immunized and boosted, you get increasing amounts of primarily non-neutralizing antibody responses. Whereas if instead one could rescue this by even if you primed with something that wasn't that great, if you were able to boost with a better kind of protein, you might get a neutralizing antibody response. Okay? So this is a strategy that we applied in the case of HIV. I won't talk much about it. Now, as I said, neutralizing antibodies are often a very good correlate of protection. And as I was mentioning earlier, enveloped viruses require surface proteins to bind to their cellular receptors. And these are typically in metastable conformations. And so we need to find ways of stabilizing such protein. And one way is to introduce mutations to destabilize the post-fusion conformation of the protein. So it's important that we know not only the pre-fusion or the native conformation, but also the post-fusion conformation. Now, it turns out that in the case of many viral proteins, the post-fusion conformations are relatively similar and easy to predict, even in the absence of a structure, of an experimental structure. Also, very recently, within the past couple of years, there have been tremendous advances in our ability to predict protein structures from sequence through algorithms such as that used in AlphaFold 2. I don't know how many of you are familiar with that. So the convergence of machine learning and artificial intelligence has really led to completely transformative advances in the ability to predict protein structures. And so, of course, besides neutralizing antibodies, T-cell mediated immunity also contributes to protection. And very often, larger immunogens give rise to a better protection, possibly because of the presence of larger numbers of T cell epitopes. Okay, so I'll just illustrate through one example from our work how we could apply some of these principles. So this is what happens in the case of influenza hemagglutinin, which is a surface protein of influenza. This is what it looks like in the native conformation, and in the at low pH in the endosomes, there's a very large conformational change. So the fusion peptide, which is located down here, then goes up and then goes into the endosomal membrane, and then that leads to fusion of the viral and endosomal membranes and release of the viral contents into the host cell. So if you look at the degree of conservation of different residues in the three-dimensional structure, it turns out that residues in this part, which is called the stalk of hemagglutinin, are relatively well conserved, whereas residues over here are fairly variable, and most conventional vaccines elicit antibodies against this region of the hemagglutinin surface protein, which is why influenza vaccines need to be updated every year. So earlier people had tried then to express just the stalk of hemagglutinin by itself in the absence of the head, but they were unsuccessful because it will always go into the post-fusion conformation. So what we figured out, looking at the two structures, was that there was a section of this loop over here, which becomes a helix when you go to the post-fusion structure. And by replacing two hydrophobic amino acids with charged amino acids, that would shift the equilibrium now to the folded states. So using this very simple physical chemical principle, we could make really almost pretty much for the first time a successful example of structure-based vaccine design. And when we made these stems, we could show that they protected mice from heterologous pathogenic challenge. So you can see here that the immunized mice were completely protected, whereas the unimmunized mice were succumbed. Okay, and so this is just to again illustrate. This is the structure of hemagglutinin, colored according to residue conservation. So in the this so-called stalk region, you can see that the residues are highly conserved, whereas over here they are quite diverse. Okay. So now just come to the spike protein, which is the causative, the spike of SARS-CoV-2. So here also, if you look at the part which binds the receptor, which is equivalent to the globular head of hemagglutinin, the receptor is ACE2, and there's a particular domain called the receptor binding domain. And a significant proportion of the neutralizing response is directed to the receptor binding domain. So very early in the pandemic, we showed that this particular protein is highly thermotolerant, and it can, when you lyophilize it, it can withstand temperatures, boiling point temperatures, and can be stored, for months at over 40 degrees without loss of immunogenicity. However, this itself was moderately immunogenic. So what we did then was to use methodology we had developed in the lab to make a stabilized version of the protein, which was only about maybe 6 or 7, 8 degrees more stable. But when you look at the titers of neutralizing antibodies that were elicited, these went up by 100-fold. So even just simple protein engineering can greatly help in the elicitation of neutralizing antibodies. And so when we made RBD based on the wild-type Wuhan strain sequence, the antibodies that were elicited also neutralized the Omicron pseudovirus. Whereas when we took the wild-type RBD, we got weaker neutralization of the wild-type and the Delta pseudovirus. But even here, you can see 2 animals, the sera failed to neutralize. But with the Omicron pseudovirus, the antibodies totally failed to neutralize. So by stabilizing the RBD of the spike protein of SARS-CoV-2, we could greatly enhance the ability to elicit neutralizing antibodies. So next, we looked at seeing if we could further improve on this. So it turns out that the spike protein has two subunits, an S1 subunit and an S2 subunit. So the S2 subunit is analogous to the stem region of influenza hemagglutinin. And if you look at the degree of conservation, you can see that it's much higher than it is for the RBD. So what we decided to do was to make fusions of, but the antibodies that are elicited against this particular domain are poorly neutralizing. So what we did was to combine both of these to make a single fusion protein, which turns out to be very highly expressed. And then we could show that this expresses at much higher yield than the whole spike protein and shows comparable to superior immunogenicity. And so we could show that when we immunized animals, so we could again lyophilize this protein, keep it for over a month at 37 degrees, and the sera elicited a whole range of viruses, including, though much more weakly, the SARS-CoV-1 pseudovirus, as well as WIV1, which is another group 1A, which belongs to the same group as SARS-CoV-1. SARS-CoV-2 belongs to the group 1B of sabecoviruses. And we could use similar design methodology to make stabilized versions of the RBDs of other sabecoviruses as well. And we could also show that whether it was lyophilized or non-lyophilized, we could get equal degrees of protection with the antigens. Okay. So spike is the antigen that is used in practically all licensed COVID vaccines. And so what we could show is when we compared our molecule with the spike, firstly, the yields were much higher. The ability to elicit neutralizing antibodies, so ours is in blue and the spike is in green, was in many cases statistically significantly better. When we looked at challenge in hamsters, the hamsters showed a lower weight loss. You can see the blue is above the green line, and there was less lung pathology in these animals. Okay. So now, knowing the structure of the immunogen is essential for vaccine design, and as I've mentioned, experimental structure determination is extremely difficult and laborious, and even with all the advancements that have been made, it can take several months to years before you get an accurate structure. So recently there have been all these tremendous advancements in AI and machine learning, which now allows us to very accurately predict, almost to within the era of experiment, structures of many proteins of interest. One area where these advances have not been so transformational is in the area of improving protein stability. So although there is some advance in the ability to predict mutations that will increase stability, this is still a very much evolving area. And the primary problem is that they have, with protein structures, because of the protein data bank, there are a very huge number of structures on which these algorithms can train. Whereas for protein stability, we don't have that many protein stability measurements on diverse proteins, and also looking at the effects of mutations on protein stability. And so we need more experimental data to improve these predictions. And I think in the not too distant future, we'll have a situation where we can use these kinds of advancements to actually predict mutations that will enhance protein stability, that will be of tremendous utility for vaccine design. So again, for want of time, I won't go through the process of how AlphaFold works, but essentially it takes a lot of data both from known structures as well as importantly from multiple sequence alignments. And the very important principle that it uses approximately is that if you look at a large number of multiple sequence alignments, there will be what are called correlated mutations. So that whenever a mutation occurs at one position, in many sequences, there will be a corresponding mutation at a nearby position. So you can see there are two yellows here, and then, well, maybe that's not the right. But any case, I think you know what I mean. And so by looking at a large number of sequences and inferring looking at the correlated mutations, it turns out very often these happen when residues are close together in space. And so that can then be used to build structures of macromolecules. There are also something called large language models. I'm sure most of you nowadays use ChatGPT or some equivalent to do a lot of your work. And what these large language models do is they look at large collections of words in their context, and then this allows one to actually construct complete sentences and apparently meaningful paragraphs just by all the training that has been done to see how words are put together in different contexts. So exactly in the same way, people can look at large numbers of protein sequences, which are available now very cheaply thanks to the ease of doing gene sequencing. And by looking at all these sequences, you can actually infer about what mutations one could do to improve protein stability. And this is because if you have the right amount of training data. So if you represent the wild type and the mutant proteins in some form, so each amino acid in the sequence is represented by a very large number of parameters. And these are often very arbitrary parameters. And so you finally get a protein descriptor matrix. And so the difference, this is one representation, one specific sequence of the protein, and this row may be another. So it's a reasonable assumption that the similarity in these two representations is related to the change in the difference in free energy between the sequences. But you have to train appropriately. So the difficulty here is getting enough training data, but this is an approach that is rapidly giving some information, but we are still a long way from this apparently simple problem of being able to predict the free energy change associated with a mutation in a protein structure. Okay. So as I said, one of the things that became readily apparent during this pandemic is that despite the very rapid speed at which vaccines were produced, they were still not fast enough. And in fact, if you look at the world map of deaths versus vaccinations, they're sort of uncorrelated. It's pretty interesting to see. So one approach that people are taking is to now do this vaccine design in advance. And by taking different classes of viruses that have epidemic or pandemic potential, and then from each of these different classes of viruses, select certain prototype pathogens for which one can make vaccines in advance and actually put them through animal testing. And so the idea would be that you already have vaccine candidates, and of course you don't know which exact virus is going to cause a pandemic, but if it's similar to one for which you already have a vaccine, then that greatly enhances the ability to make a new vaccine. And this is actually what happened in the case of COVID. COVID vaccines didn't emerge from nowhere. So this is the structure of the coronavirus spike. This is just a human coronavirus, not SARS-CoV-2. And then what researchers, Barney Graham and his Jason McClellan and their colleagues discovered was, first of all, this protein was not very well expressed. Both this as well as the MERS, which is the Middle East Respiratory Syndrome coronavirus. So they found that in a particular loop in the protein, if they introduced 2 proline residues, they got a very large increase in the amount of expression. Okay? And this very interestingly goes back to seminal work that was done by G.N. Ramachandran many, many years ago in Madras University. And what they've, what some of you may or may not know is these are the phi and psi dihedral angles which describe the conformation of a polypeptide chain. And because the proline, as it turns out, it has a cyclic structure over here. And so that results in a great restriction in the values that the phi conformational angle can take. And therefore, because this has a restricted dihedral angle, when you have prolines in the unfolded state of the protein, it restricts the number of conformations that the unfolded state can take. And as long as those prolines can be then accommodated in the native state without significant energetic penalty, this can lead to stabilization of the protein. And so, though I'm not sure they quite understood it in these terms, but anyway, that's what they did. And because this had been done in advance for the MERS and as well as the SARS-CoV and the human coronavirus protein, when SARS-CoV-2 came along, which was a very similar kind of protein, even in the absence of a three-dimensional structure, they were able to very quickly make these mutations and then make mRNA, which could express that protein. Now, even the mRNA vaccine work did not come out of nowhere. Okay? So if we look at, these are all slides that Barney Graham kindly gave me. So very early on, well, a few years ago at least, they decided to develop this prototype pathogen approach for pandemic preparedness, some of which I briefly described, and they wanted to propose 3 different mRNA vaccines and compare those with DNA vaccines. Then there was an agreement to do this between the Vaccine Research Center in the NIH and Moderna. And then in August 2018, they proposed, again, another prototype pathogen, which was Nipah virus. In September 2019, well, just before the pandemic, as it turned out, they got into an agreement with Moderna to see the utility of mRNA as a vaccine modality and to have a pandemic response demonstration project. So they decided to go ahead with, a small batch manufacturing for a phase 1. And then, very quickly, when the actual pandemic happened, and because of all their prior work that they had done on the other coronaviruses, they were able to move this forward extremely quickly. So you can see that there's all this preparatory work that went on for the previous 5 or 6 years, and even longer if you take into account all the advancements in structure-based design. And so when the respiratory outbreak came out, they realized in early January that it could be a coronavirus. The sequences were soon published, and then in 3 weeks, they made the spike protein, which you need to make protein to do potency assays and so on. And then they could look at the, make the mRNA, get antisera, look at cross-reactivity. In the meantime, the spike structure was solved. This was not necessarily such a huge advance because the SARS-CoV-1 structure was already known. And then, within within about 2.5 months, they had a phase 1 trial in progress. And the reason this happened is because unlike protein manufacturing, every protein is different. And so the regulatory procedures and processes that are required to approve a protein vaccine tend to be much more time consuming. In the case of mRNA, because the making one mRNA is as easy or difficult as making another roughly similarly sized mRNA, all these regulatory procedures could be somewhat bypassed. Okay? And also the toxicity of the mRNA also is not completely, but less likely to differ from one mRNA to the other. So they were able to start a phase 1 clinical trial very, very fast. Okay? So you can contrast this to the situation in India where, again, we had the first immunogenicity data for a protein subunit vaccine formulation in about April 2020. And to this day, we have not received regulatory permission to make the protein, to put into humans. Okay. So how can we, this may be not exactly immunology directed, but I think it's useful for students to know and appreciate. So how can we improve our future responses to these threats? Because now, of course, COVID is over, everything is all forgotten, and I don't think anything will be done. But it's worth keeping all this in mind, because really we were fortunate, I think, that COVID, as bad as it was, the infection fatality rate was not particularly higher than influenza. So, there were a lot of deaths, and it's very sad. But if something like avian flu comes into the human population, there'll be complete mayhem. So there were many, many lacunae that became very apparent during the pandemic. And, of course, we need broad-based funding so that there is a sufficient pool of expertise. I'm sorry. Okay, we need better surveillance. And one of the things that became very clear when we were working during the pandemic is that there were no reagents available. Even elementary things like gene synthesis couldn't be done at the time in India. The situation is a teeny bit better now, but not a whole lot better. And, we couldn't get approvals to work with the virus, even though, I think it took almost 8 or 9 months before we actually could get hold of the virus, though by that time, 100 million people or more had been infected, but the virus was so closely held that it was impossible for, except some very privileged few to access it. Yeah. And we were not allowed to do any work because there was a lockdown. So, okay. So I think, let me just conclude. So there have been a lot of transformational advances that have greatly accelerated vaccine development, but there's still a number of important challenges that remain. And this is, I believe, one of the rate-limiting steps is finding the right molecule that will elicit a protective immune response or the right formulation. And we do have a lot of strengths, but unless policies improve, it's going to be difficult to move forward. Okay, so I'd like to thank all the people, in my lab and my collaborators, as well as at Minvax, the startup that I co-founded a few years ago. Some of the slides I got from Barney Graham and from my friend Madhu in ISER Pune. And this is the funding. And thank you.
Sir, I wanted to ask that, in non-replicating virus vaccines, the expression of the gene of interest which we have inserted through the viral genome insertion. So, sir, is it that the protein which is made is in the right folded conformation? Like, once you insert the gene of interest, like
Correct.
whatever protein you want to make. So how is it possible that, because it is making at the cellular level inside the cell, so how do you get that folded conformation that you want?
So it's being expressed in the same way that the viral protein would be when it was expressed inside cells. All the same signals are available. So the assumption is that it's expressed in the folded conformation, but the only way really to check is to see what the immune response is and see whether you are getting the right kinds of antibodies. So there's no way, so that's, that's what happens with mRNA vaccines as well as with the viral vector vaccines. You have no way of actually doing any of this characterization, which is why I think the regulatory pathway is paradoxically much simpler. For a protein subunit vaccine, the regulators want all kinds of excruciating details, including, which glycan is on what the kind of glycan is it folded, is it not properly folded, how heterogeneous is the material that you're making. But with mRNA and viral vectors, there are no such requirements because they can't be done. So,
Okay, thank you, sir.
yeah.
Any other question?
Anybody done this work that adenovirus vector spike protein? When they actually come out of the cell, this protein is maintaining this structure really?
Well, that was the question. But you can't, the amount, it's all produced in vivo. And the amount of protein that's produced is too small to characterize. So, and quite ironically, although it was known that these two proline mutations are important in maintaining, making sure that the protein is in the right conformation, they were not incorporated into any of the COVID vaccine, the adenovirus vaccines that practically all of us have taken for no good reason, as far as I can tell, because this was all published data. So, and even to this day, they have not been incorporated. So, yeah.
So, proline causes a kink in the protein structure.
Protein causes what?
Proline causes a kink in alpha helices. These
Yes.
were put in a loop region. So it doesn't really change the structure of the protein, but by stabilizing destabilizing the unfolded state enhances the stability of the protein, because the stability is the free energy difference between the folded and the unfolded states. That's how.
Thank you.
Yeah.
Back there.
Thank you for your nice talk, and it's very interesting to see the proline things come, because this is the strategy of many of the membrane proteins, especially which I have worked on, that proline is present immediately before the active site residue, one or two active site residues, so that it can remain static and focus on the binding.
Yeah.
But I heard twice about the AlphaFold or such algorithms, but I don't know exactly how many numbers have been done and checked. But there are examples where a single mutation causes a very, very large change in the structure, which is very, very less possible picked up by these algorithms, because there is no, as you said, I think twice again, that a good learning data is required. So there is no much data on that. There is one example of the lactose permease, especially this is focused on the membrane proteins, where a single mutation of the glycine changed the conformation from inward open to outward open. And that kind of
Sorry,
change.
I'm going to restrict this to asking questions rather than going into such a detailed description.
I got the point. I can briefly answer.
Please respond, because
Okay.
otherwise we'll do it outside.
Okay, so the thing is, I think what should be emphasized is, so AlphaFold is not designed to predict effects of mutations or stability changes of mutations yet because of paucity of data. But what it is very good at doing is to predict conformations of naturally occurring sequences, because of the availability of homologs and the availability of these correlated mutations and what is known about many protein structures. So they're not the same problem. One is predicting the structure of a naturally occurring sequence, the other is predicting the precise structural changes that occur in response to mutation. So what it's doing is the first one.
Any further questions? I don't spot raised hands. Okay, with that we will thank Raghavan and move on.
Thank you, sir, for that knowledge-driven lecture. I request, yeah, it's okay. I request ma'am to provide the token of appreciation to sir.